audio.
Olive oil for 6 months, not like, yeah, not a thing.
Very crunchy.
Hello.
Professor G, do you want to try saying something so we can see
Hello.
pick it up well?
Why don't you stand here and say something
Hello.
and pretend that you're me?
Hello.
Okay.
Hello.
Testing?
Do you hear yourself?
It's good.
Alright, cool.
It's picking up pretty clearly.
So, just to be clear, let's see on here, I don't actually need to see myself, but. So hold it to about here.
We can move more on the board.
We might
can
just
also
move the
just, like,
table back.
shift it or move it back.
No, I think that's okay.
Perfect.
That's good.
And is it the right orientation?
Yeah, it should be the right one.
Is there high enough resolution that you can actually see it?
On the screen it looks like you could read that. But I think you'd have to write always that big-ish. Anything smaller might be a little rough.
Okay. But you said streaming now?
Yeah, it's reading now.
Is there anyone online?
Let's check.
There's two.
Two. Is that you and Michael?
Could be. Could be. See if we can just jump into it.
Sorry.
See if we can-
You can also refer them to Sunny's Google Doc, and I think Miriam is also taking a backup Google for
Yeah.
some kind of document. Turn it off at one. Just turn off lights in another room. Hey, that's awesome. Yes, I did. Look. Oh, it's okay. No. The keys that were in here. You still see that if you like that?
I think you should be okay. I think I'll probably for most of these questions, if there's a lot of writing, I'll type it up in the chat as well.
That's up to you guys. I don't know
It's
what
pretty
you.
clear, but like just in case.
Okay, and there is like a Google Doc, etc. So you can put that directly into your
Sure, yeah.
YouTube stream or whatever.
Paste the link in, yeah.
Okay, so just a couple of really quick things before we start the Q&A, sort of open office hour / review session. Happy to cover anything that you ask. So I've been asked this like 3 or 4 times since the end of the course. The final exam is not cumulative. It's in your syllabus. I've mentioned it several times. It's everything from the last term test. It's 15 multiple choice. 10 one-line responses, similar to what you had on the last term test, and then 3 short answers, which are basically about a page-ish in length, but I'll get to that in a moment. These are all worth 10 marks, this is worth 10 marks, this is worth 15. In the past, and again, it's just a ballpark figure, the multiple choice average over the last few years has been about 12 out of 15. The one-line responses, again, on average have been around 8, sometimes a little higher, sometimes a little bit lower. The short answers, usually, depending on the year, is somewhere around 23 to 24, so we'll ballpark it at around 23.5. And again, it depends a lot on how you write your short answer questions out. And over here on the left is an example of a short answer question, which you would get like 1 page, and I'll get to that in a moment. Hey, Kai, how's it going? That's sort of the plan. The way that I would sort of look at this is a 2-hour-long task. And again, this is ballpark, so you should just take it for what it's worth. Like for the 2-hour time that you're allotted, you're given the full 2 hours, so remember, show up on time. It doesn't start at 10 after, it starts at exactly on the hour. In fact, you'll get a whole bunch of instructions before the hour starts. So generally for each of these short answer questions, you should be kind of budgeting around 20 minutes-ish. You're not supposed to overwrite. So that means that this section here, with review and everything else, please plan your question in advance. Like you're not going to be given an extra sheet. You can write in pencil if you want, but once you start writing, like you're committed to filling out that sheet. You're not going to be given like another test paper. So this is going to be about 60 minutes. On average, this takes about 12 to 15 minutes. Depends a lot on your English facility, how comfortable you are with multiple choice. And then this also takes about 10 minutes. So if you add all of that up, it's about an hour and a half. That gives you about half an hour to review. So some of you will be done in about an hour and 10 minutes. Some of you will be done exactly 2 hours. But on average, if we're looking, it's about an hour and a half exam if you like spend time reviewing and everything else. Does that make sense to everyone? And that's sort of a ballpark that you can use like when you're looking at the exam. Like, wow, I'm like really behind and I should be like budgeting this. Some of you have different strategies, which I can't comment on because it should be something you're comfortable with. Some of you start the short answers first and then you go back to the multiple choice. Some of you start the one-line responses. It really depends on your comfort level, but that's how I would kind of outline it. Do you have any questions on any of this before we go on? No.
This is a silly question, but if you misinterpret something in the question and you make a mistake very early on in the question, but the rest of your logic follows through with that misassumption, so like you said, the wrong protein or the wrong pathway, but then your answer goes with that pathway, do you get like any marks for having that part correct, or is it wrong just because you started wrong? Like, will you give part marks or partial marks?
Okay,
for
so let's use that short answer. I'll come back to your question.
Sure.
So generally, as a default, Miriam and everyone else, I don't like giving 0. So how's the best way for you to get a 0?
To not answer.
Not answer it. Leaving it blank, you get a 0. So I think that in general, I am very adverse to giving like zeros on any kind of written question. So if you started off and you totally misinterpreted, like, holy cow, I think AKT kinase is a enzyme that phosphorylates Krebs for whatever reasons. And then you started off on that pathway, and then you got into like Krebs, phosphorylation, and got into all of that. Technically it's wrong, but you wouldn't get like 0. So you may not get full marks, but you won't get 0 either. So it would depend a lot on things that you included in your answer. And if you look, the answers are kind of compartmentalized anyways. So they're designed so that you can't actually get 0, like unless you just don't try. So like even if you didn't know this, you could still answer this part. Like you could still answer like, I know how to test for anxiety. You know, you go show up to a U of T class, and then you sit there, and then you take the final exam, anxiety. But you can like compartmentalize it that way. So you should never approach a question like, I'm gonna get 0 1. You should always approach it like, take a look at the question. Many of you use the strategy of like underlining keywords, all great. So like, hey, I don't know everything right now. I'm gonna start writing and planning it out, that I'm gonna start with like anxiety, test behaviors, and that's how I'll begin, right? Okay, any other general questions before, yes?
Can you answer those in point form?
Yes, you can. I'm happy with it in point form. The only thing I'm gonna mention, and we'll get to the short answers in a second. So, oh, that's gross. There's like hair on this question. Yeah. Okay. You guys will have to let me know if there are any questions on that. Thank you. Okay, so the way that the test questions are organized, so this is your sheet of paper, and then this will be like question number 26, for example. So it's 26, 27, 28. And then you get like this block of text up here. It tells you that this is worth 10 marks. So you know that each of the short answer questions is worth 10 marks each. And then you get these lines and a couple of really common misconceptions that students have, just to make this like really clear. And then it says do not write in this space, right? So a couple of really common misconceptions. You don't have to fill this to get 10 marks. Like that's one thing that students always feel like, hey, I gotta fill this whole thing up for 10 marks. Not true. So what I'm usually doing is looking for keywords that you understand the question, you've understood things from lecture, you're able to apply it, and all of that. So can you use bullet points? Sure, I'm happy with bullet points as long as they cover all of these things here. And you can like use bullet points. The thing that I don't want you to do is like for each of these lines that you have here, don't write like here and then here. Like students will do that to kind of use space more effectively. If I see that and you continue to do that, like I'll deduct marks. So don't do that. Just like write as you'd expect. Most of the time I'm kind of expecting if you have normal size handwriting that you're kind of leaving a couple of lines here at the end, and that's pretty much it. So try not to write really, really small. So if I can't read it, I can't mark it. And you might think that it's legible, but it's like so small that I can't read it. It's just not going to be something that I can grade. Usually I'll make sure that it isn't something that I'm being biased against, so I'll ask one of my colleagues, can you read this? And if they can't also see like your really, really small writing or it's really messy and we can't interpret it, then we're just going to try to read it the best we can, but if we can't read it, that's also something that we just can't help you with. So I'm also asked this, because this came up quite a lot. So it says, please do not write in the margins or in the spaces. So like if you have a couple of words here, do you lose marks? No, you don't lose marks. Try not to like do that for every single line though, but if you have a couple of words here, that's fine. Like I just don't want like people to write double spaced, and that often happens. One year I had students writing 3 lines for each of these spaces, and it was just like, why bother? So don't do that. You'll lose like 1 or 2 marks for each of the questions that you've done that on, so try to avoid that. Just read the question first. Use the space here up at the top to like, I think I'm going to need to talk a little bit about like apoptotic pathways, or I'm going to have to talk about like different proteins downstream, and then plan it out and then sort of get your thoughts in order. You might want to think a little bit about what kind of anxiety tests have I learned about, so freezing, startle responses, light-dark chambers, 0 mazes, whatever you remember, and write those down at the top, and that's how you get started. But I would literally spend like 1 or 2 minutes kind of just planning it out and seeing what I remember, and then kind of mapping it out here. You can write in pencil, like you're not required to write in pen. So you can always go back and erase it, but if you're writing in pen, you should bring your whiteout or whatever else. Anything else that I can answer? I don't know if that answered your question. Anything else I can answer for you? Yes?
Will you be rounding down our final grades?
What does that mean, rounding down?
Like I know that sometimes like classes have like too high an average and they need to like ramp down people's final grades.
No.
I know,
So do you mean like I have
Like,
I have an 84.9 and I rounded down to the 84 because that's the last whole digit. Like I know some people do that.
Okay.
Yeah, I don't.
Like some files are worse than that, will actually like bell curve down
Yeah,
instead of
that's
bell
what I'm
curve up.
No.
Like
That's
doing
right.
the whole thing, is just, yeah,
Just a quick question, when are we getting our literature review?
I think it depends. The TAs have been kind of looking at them. I've been looking at my section. I've been uploading the rubric and putting the rubric online for students so that you can actually go back and see like what you got out of 4. I don't know how other TAs are doing it. I'm only about I've only marked about 10 to 15, so I still have about 20. I still have about 35 more to go. Lauren, did you have a question?
The only other question I'd ask, because you mentioned the whiteout, is it cool to just cross things out?
Yeah, I think you can do that, Lauren, but you're try not to like write too much up here at the top.
Right. No, I just mean like cross it out and then just keep writing, not.
Sure. Yep. Yes.
It's more on content.
Okay, are we going to content now?
Is that okay?
I think that's why we're here. Okay, we're gonna try to do it sequentially. So we're gonna do it from the first lecture after the term test, and then we'll kind of go from there. If you have any questions about content, anything like in general, like you're free to ask. Like use this as open office hours, wherever possible. I'm happy to answer like any question that you have. So feel free. So first lecture after the term test. Do you guys have any questions from your notes, or are there any questions from online? Any questions? What was that lecture on?
Depression.
Oh, okay. Very bright topic. Any questions from that lecture?
Yeah, I just have a question. Sorry, I didn't check the answer online. But I was wondering if throughout the lecture you kind of said that it was one with CBP and
Right.
only admits
Right. So in the very last lecture, on slide number 2 or 3, I went back and answered any unresolved questions. And one of the things that I mentioned in the last lecture, so I think slide 3 or 4, that when we look at the cyclic AMP response element that drives transcription, so different genes would be turned on, that one of the things that, including things like BDNF, for example, that we require like the dimerization of Krebs, and usually we look at phosphorylated Krebs, and this gets to serine 133. And I had made an error earlier talking about Krebs binding protein, which also kind of sits in this complex, but we didn't talk about that. So it's Krebs serine 133 that gets phosphorylated.
I was also just out of curiosity. So you also do need to activate CBP, right?
In most cases, we activate and phosphorylate Krebs, and we also have accessory proteins like Krebs binding protein. We have that whole tetrameric complex. But that's not something that we covered in lecture this year. Yes?
In that diagram, it showed 2 Krebs.
Yeah,
So does it
It dimerizes.
both of them, like come together to make,
That's right. It's a dimer.
and then CBP comes and attaches?
Again, you don't need to worry about those details around CBP because this year we didn't really cover it. It's really about the dimerization of Krebs binding to the Krebs response element or the cyclic AMP response element, CRE, and then causing transcription. So in previous years, we've covered in more detail like the CBP, accessory proteins, but we didn't this year. Okay, so that's not really a lecture, whatever. Any 5. So any questions from lecture 5 on depression?
At one point you said substance users show signs of depression.
Yeah.
But like isn't that an increase in dopamine, so why is that?
Yeah, so there's so is it Sarah or Marwa? Marwa. Okay, sorry, my apologies. So I think in general when we look at substance abuse disorders, we are talking about enhancement of dopamine through the different substances that they're using, but their brains under normal conditions when they're not using different substances actually have a depletion of things like dopamine and a depletion of other neurotransmitters.
Because of taking medicine?
Again, there's a lot of controversy about that. Not every substance abuser will have like depression, not every individual will have that, but that's the hypothesis that it's like through overuse of the system or downregulation of some of the pathways. Yes.
There's also the argument, I don't know if it's relevant to this course, but like that people self-medicate with things like substances, so like maybe is there possibility of there being a like pre-existing depletion of certain hormones that lead them to seek some sort of like, you know, reward they would get from like alcohol or substances? Is that also a possible?
Yeah, there's a so without getting too sidetracked, Miriam, there's a very controversial field within neuropharmacogenomics where some individuals believe that certain genes are actually more highly regulated or inactivated in different substance abuse disorders. So you'll often hear about the DR2, the dopamine receptor 2 gene being implicated in different forms of addiction. Again, very controversial. Some people think that it's a real thing, that yes, you have like a mutation in the dopamine receptor 2 that leads you to be more likely to engage in substance abuse, and some people feel like, no, that's not true. It doesn't matter if you have that mutation or not. Yes, Laura?
There's also a speculation to the same thing in opioid receptors.
Yep, 100%. So I mean, it's very speculative that different types of mutations or truncations will lead to different types of substance abuse disorders. But again, it gets very, very controversial, right? So that means that you're genetically predisposed to being a substance abuser, and you know, that's not an easy label for someone to have. So, other questions? Dopamine notwithstanding, lecture 5. Yes.
You briefly mentioned something about anterior strokes near the CG25 area resulting in depression. You expanded a little bit on it, but I just wanted to know like, what you should know. Is it just like one of the like side effects of the stroke in that area? Is that just?
So I don't think, I could be wrong, so correct me if I'm wrong. So I don't think I actually mentioned area CG25 directly on that slide. So I think what we were talking about was really like the norepinephrine system or the noradrenaline system. Okay. And we were talking about specific groups of neurons, like the locus coeruleus neurons, and that we have like within the brain, we have very, very discrete groups of these noradrenaline producing neurons that then send their projections throughout the brain. And that if we have, for example, an anterior stroke that will target this pathway here, that this could lead to depression. So we're targeting this particular pathway. The white matter tracts that deliver norepinephrine to the rest of the brain, they're not going to be functioning anymore. So it means that there's an implication that norepinephrine, at least anecdotally and correlationally, so it's not direct evidence, but it's correlational, that this anterior stroke in this area, specifically this area, could cause depression. And that implicates the norepinephrine or noradrenaline system, which is how I believe we segued into that topic. And again, correct me if I'm wrong, because it's been a while for me too.
Okay, that explains it. Thanks.
I think you might have to do it close to CG25
Yeah,
area.
so the one thing for sure, so like area CG25 is like implicated and has been implicated in some very severe treatment-resistant forms of depression. And this area does indeed become very active on an fMRI for individuals that have depression, or for normal individuals who are thinking of things that are really sad. Any other questions? Yep.
This is kind of a silly question, but can you just go over Dr. Fox's again a little bit and just explain how that works with the reward circuits? You talked about, I think that was the last couple of slides.
Right, okay, so as we segue into your question, Miriam, so that gets into like this question. So I'm specifically asking here, like which groups of neurons, like so you might be thinking about the different types of neurons that we discussed. Not necessarily in this question here, but in that specific context. So we know, for example, that we have one system where we have like delta phos B. This is what you guys learned this year, right? Phos B. So we know, for example, that if we have certain circuits in the brain, and you have to think a little bit about this because this is like lecture 5, right? And then we went all the way to lecture 10-ish, is that right?
Yeah.
Before we get to the nucleus accumbens and the VTA pathway, which is what we concentrated on. Before that, do you remember another circuit that we talked about? There's another place where delta phos B is like very highly regulated. It's like very highly expressed. Right, so do you remember periaqueductal gray from your notes? Vaguely. Okay. So periaqueductal gray is a neural circuit that you actually kind of know. So I'm going to tell you in advance, like I like your class enough not to do this to you, but in theory, like you've learned periaqueductal gray in lecture 10. Do you remember that one too? Yeah, so this is sort of the default pathway that we use for placebo. Do you remember that one? So we start with the dorsolateral prefrontal cortex, we go all the way down to periaqueductal gray, et cetera, et cetera. So in theory, like that would be an excellent example of where I combine like two different elements, periaqueductal gray, delta phos B, and then also related back to what we looked at for placebo. I promise you I didn't do that, but that would be like a really prime example of like applying it over multiple lectures, okay? So you don't have to worry about that for here, but just keep that sort of running in the background. Okay, so does anyone remember, is this upregulated or downregulated in resilience?
Upregulated.
So normally, in mice that we see being resistant to depression, right? And you might want to start thinking, okay, what models do you know for testing for depression? You know a few. In fact, you know more than a few. Do you guys know what you would use? Okay, so we would use, and by the way, these are perfectly acceptable on your answers for swim test. Just write FST. Anything else? So we could do tail suspension test. Right, so here it's a little harder to write out, but sucrose preference test. If you're like really clear about it, you could write SPT, but just be like really clear that these are all kind of tests that you're using for induction of depression. So when we look at these different models, and that's how we test for, how do we induce depression?
Social isolation.
So we could do social isolation. Anything else? So we could do chronic unpredictable stress. We could do like random foot shocking, all of those things. So there's a difference between, and again, be really clear on this because you might need it for your final exam. These are models that we use to induce depression. These are ways to test whether or not depression has occurred. They're not the same. Okay? And often students have them confused. They're not the same. We use these to induce depression. After we've done it, if we have induced depression, we will see less movement on a forced swim test, we'll see less struggling on a tail suspension, and we will see less sucrose preference versus like water, right? Okay, so if we overexpress delta phos B, at least according to Eric Neffler, one of the things that we would expect to see is that we have less depression. They're more resilient, in other words, right? So they're more likely to continue struggling. And in particular, we would overexpress them within the reward pathways. So particularly within the nucleus accumbens. And this is where we talked about some of the data for the expression of the gene. And we saw that there was more in the nucleus accumbens shell, I think, as well as the core. So in both areas. we saw an increase in delta phos B in animals that were resistant or resilient. Does that make sense to everyone? Okay, so one of the ways that we could go back and test this, and there are lots of different ways that we could test this, if we see that this is overexpressed in resilience, we should actually expect as a hypothesis, which again gets into like hypothesis testing, which is like the new thing as a learning outcome for all of you in your third year, fourth year. So for hypothesis testing, you would say that if this is important, if we reduce these levels, they should no longer be resilient, right? And one way to do this is to actually antagonize the binding of delta phos B, which is a transcriptional factor, with delta C Jun D. So normally we have phos and we have C Jun. These come together as the AP1 complex, but the delta C Jun actually acts as what we call a molecular decoy. So it takes and instead of making it transcriptionally more active, it binds it up so that it can't actually work with the normal C Jun. So it's using it in place of the regular C Jun. So it's like what would be a good analogy? So for example, I'm asking you to write something like in a specific blank on a sheet, but if I have like a whole bunch of blanks, like you're going to try filling out all of them, right? And so you might not fill out the one that I'm thinking that you're going to fill out. So in the same way, if we have like delta phos B, we want to make sure it's not binding to the appropriate region or not being filled in to the proper gene sequence. We can just saturate it and bind up all of that by just giving it more sites for it to bind, that it won't work appropriately. So it's called a molecular decoy. Any questions on that? There was one other way that they used to kind of knock this down. Do you remember how they did that? So one way is we can bind up like the delta phos B, and they do that. They overexpress C Jun D. Their levels of struggling, they go back down. What else did they use? I can't remember if they used RNAi. That would certainly be acceptable. So you could use like RNAi. I don't think they used RNAi, did they? In this experiment? I don't think so. Did they use like antisense oligonucleotides? No one studied lecture 5.
Yeah, I think so. But
Does anyone have
I had it
lecture?
open, I don't remember that being talked about.
Going off camera. Wait.
We did the ABC, we can have both in that way.
They didn't do that. Okay. So
Yeah, we did, that sounds really.
so just going back and reviewing the slide. So one thing that they did is they used the virus to overexpress delta phos B. So when they do that, then the animals are more resilient. And then they used like the delta C Jun D to act as a molecular decoy, reducing the levels of delta phos B. So that's how they did it. So if you use something that we didn't talk about in lecture as part of that response, and it made sense, like RNAi, would we accept that as an answer? Yes, of course. But as long as it's logical, then we'd accept it. Anything else from lecture 5? Yes, Lauren?
Can you explain the difference between delta CGMD and
C-Jun
yes.
and just C-Jun. Right, so normally you would have learned about like C-Phos and C-Jun at some point in your lectures in like first year, second year. That can't be true. That can't be true.
Never.
It can't be true. You're supposed to learn this both in your first year bio, Phos and Jun. Immediate early genes. You've never heard of immediate early. You're supposed to have learned this in your second year neuro. Immediate early genes. So C-Phos and C-Jun make up the transcriptional activator AP1, and this turns on when you get Phos and Jun together. They will form this complex known as AP1, and AP1 just turns on a number of different genes, including like BDNF and a number of the other ones that are on that slide at the bottom, and that's why we use it. So delta C Jun D will bind up any of the delta phosphate. So phosphate is like analogous to CPhos, and C Jun D is normally what it would bind to. But if we have the delta form of it, it binds delta phosphate, but it doesn't allow it to form this AP1 complex. So imagine for a second, well, I think I'm getting really off topic. So if I'm getting too off topic, let me know.
I've never heard of this stuff. Have you guys ever encountered this stuff? Okay, I just feel like I'm maybe the only one who doesn't understand this. Okay.
I think we should go back onto the next lectures, because we only have one lecture.
We can look it up later.
No, I have to answer that question now. So you have like different transcriptional elements that you can have like binding, like promoter regions. You know about those, right?
Yeah.
Okay, great, fantastic. So one of the most common type is anytime you like electrically stimulate a neuron, anytime like you add a neurotransmitter, like there are small amounts of like CPhos being turned on. Like the neuron knows something's happening to it, so it turns, like if you touch a neuron, for example, it will upregulate CPhos. And the neuron has to do something with all of that stimuli. So CPhos, C-Jun, they come together. CPhos is like upregulated by itself spontaneously in terms of any kind of increased activity. CPhos and C-Jun will like bind together to form AP1. So similar to like the Krebs phosphorylation dimer. So it's a common theme. So these two things come together as a complex. AP1 then turns on like BDNF expression or other expression. Okay, so going back here, so we have, at least in these neurons, we will have a lot of this interesting protein called delta phosphate. So it's analogous to this one here. Yeah, like CPhos. And we know that the more of this delta phosphate that we have, the better the neuron likes it. The more the neuron is going to be resistant to depression. So you have like lots and lots of delta phosphate. In theory, the neuron likes it a lot, right? So if we overexpress delta phosphate, the neurons make the mouse more resilient. So one thing that we can do is that normally this would bind to C-Jun D. So this would bind as well. These would then come in, form an AP1-like complex, cause this to be transcribed. But instead of that, let's use delta C-Jun D. So some of this will still find its normal partner C-Jun D, and it will come and activate it. But if we provide a lot of this C-Jun D here, it binds it just like it normally should, but it doesn't allow it to activate this promoter region. So by using something that looks similar enough to it, it kind of fools the neuron to kind of not allow for transcription. Okay, so if no other questions there, on to lecture 6.
There was a slide showing pictures with two mice where that's one that has the lens.
Yep.
Yeah. Could you explain that experiment?
Yep.
This is in
Lecture 6. Okay, so Lecture 6, neurogenesis, right? Right. So we have a lead shield. So you don't really need physics to understand this, but you know like lead blocks high radiation or highly energetic radiation, right?
No.
You know when you go to the dentist and they put this lead shield on you, it's to prevent like any of the X-rays to get in there into like the rest of your body. So what we do is we put the mouse head under like this lead shield, and we align it so that it's right near like the dentate gyrus. Do you understand why the dentate gyrus? Neurogenesis, right? We have dividing cells, right? Have I lost anyone? We're good so far? So you know that there, at least in the mouse brain, there are only two confirmed areas where we have like neurogenesis. One is the subventricular zone, the other one is the dentate gyrus of the hippocampus. And so we put the mouse head, so the mouse's nose is here, whiskers here, ear is back here, the hippocampus is right there, and so we have it under like the light shielding. Then we hit it with like really high energetic radiation, so we will use things like X-rays and we'll bombard it. But the rest of the mouse, like this part, it's shielded. Only this part is open. And so you have to think a little bit about what that means. This whole area of the mouse has been irradiated with highly energetic radioactive particles. And so any dividing cell, we are going to cause DNA strand breaks. They can't divide anymore. And some of those different cells are the ones that are not in the brain, but are part of the hair. So the hair follicles, they normally undergo mitosis as well. And so they turn white because the melanocytes and the hair follicles, they're the ones that normally have DNA dividing. So like your hair normally will keep its color until you get older, the mitosis stops, and then the melanocytes stop producing the coloration. So that shows you that they've been irradiated. That's really all the white strip shows.
So basically, because it's not the dentate gyrus, then there's no more.
Right, so that is no more neurogenesis, and we know that it has probably been affected because it's blocked the melanocytes that normally produce the hair color. They've lost that ability, so the cells under it have probably also lost it. The rest of the animal's head is protected, so that's why the hair color stays the same. Okay, I feel like we're covering so many different things. Yes.
Sorry, from online,
From online.
going back a little bit, but they were just wondering in terms of all the discussion we had about delta C, gene D, and everything like that, would the specific neurons that they affect in the nucleus accumbens be the serotonergic neurons, or are just general neurons?
Wow, that's such a loaded question. Wait, so you guys have all taken neuroanatomy, right?
Next semester.
Next semester.
But you know, we talk about the core and the shell. So the vast majority of these neurons that you have, like in the nucleus accumbens, are not really going to be serotonergic. So mainly dopaminergic and like glutamatergic neurons, a couple of GABAergic, but the majority are like dopamine. Okay, so that's not what I mean by which neurons. I would want you to anatomically identify them. So if you're talking about this, in that context, I would say talk about the nucleus accumbens neurons or the dopaminergic neurons in the midbrain, whatever allows me to see there. Did you understand that? Okay, lecture 6, anything else?
Can you just clarify where it's neurogenesis? So the dentate gyrus in the subventricular zone?
Yep.
What about the subgranular zone?
Still dentate gyrus.
Okay, perfect. So can you say
So,
either?
no, you can't, because the subgranular zone is just like it's just like saying you go to university, but then to be more specific, you go to the University of Toronto. Does someone at Ryerson go to university? They do. So the subgranular zone layer, it's a layer, but it can exist in a number of different structures within the brain. So to make it more specific, you would have to say subgranular zone layer. Again, this is a perfectly acceptable short form because I've used it in class. Within like the dentate gyrus, also acceptable, like I would take that. So I would know that you know that in the subgranular zone layer of the dentate gyrus, we have neurogenesis. We also have like the subventricular zone layer, and there's only one subventricular zone layer. If you have a ventricular lining, but the subventricular zone is like common to all of those regions. I don't know if that answered your question. Okay, yes.
Just at the end of this lecture, you speak about 3 different pathways. One of them is the phospholipase C gamma. Do you know much more about other than the fact that it just produces intracellular calcium?
It's not just intracellular calcium though, so wait, Miriam, why'd you laugh?
because I remember looking over this and.
Okay.
Yeah.
Okay,
So not sure what this pathway does.
So I wanted to just maybe operationally redefine that a little bit. So phospholipase C gamma is like one of the pathways and that get activated. I told you that upstream of this. So again, this gets back to like, oh, what other pathways might be involved. So in the context of what we're talking about, you're probably more comfortable thinking about it in terms of like the dimerization of the receptor for a neurotrophic factor, right?
Like any like track or something like that.
Right, so it could be like track B. We get it dimerized, we would see phosphorylation of the intracellular tyrosine residues, and then this in turn, this binding, and we'll use the most common one, we'll use like BDNF. This in turn, actually to be proper, so mature BDNF. So we would actually see the activation of like phospholipase gamma. I think we talk about it this way too. PI3 kinase, AKT. So that gets here, so you're thinking about this. And then we would also see like activation of like MAP kinase pathway.
That's also RAS, right?
Right, so this is like RAS, kinase, etc. So in this context, like what else should you know here? So from this pathway, like AKT and we are blocking apoptosis. And we talk about a couple of proteins like caspases, I think, but you don't need to know those details. Phospholipase C, you're correct. Calcium is one of the pathways. Like IP3 is like another pathway. This is the one that you should concentrate on though. Calcium. Okay. Because again, think about it the way that I want you to think about it. I'm not doing this to try to make it more challenging, but without making, without really knowing if you've understood these different pathways. So for example, you might think of it this way. So this would be a really good example of like an application-based multiple choice test question, right? So which of these different pathways might actually cause like an increase in intracellular calcium? And it may not be like this, or it might be. So it might be something like, oh, you activate track A. So we haven't talked about it here, but would track A do the same thing? Sure would. It would do the same thing, same pathway. Would like contraction of skeletal muscle do the same thing? Would it increase intracellular calcium?
No. Yes. No.
Okay. Do you remember the lecture on exercise?
Yes, it would increase.
So, again, thinking like putting it together like on multiple different lectures, like that would be an example of an application-based multiple choice, and you have about 5 or 6 of those where you have to think across multiple lectures. Like, where have I seen this in like this other context? There are none of the above or all of the above. Like, you literally have to work your way through it. Like, there's an answer in there somewhere, and you have to be able to figure it out. So that's a great place to talk about. Okay, so lecture 6 still. Any other questions?
I think IP3 is one that uses the other one is DAG.
Right, so yeah, I didn't really go into all the details surrounding like the G protein coupled pathways, the activation of DAG, etc. But this is the one that I really emphasized to your class. Like this, there is other pathways that get activated as well.
One more question. I'm not too sure what structure is. It's the one about M1 and M2.
Yep.
I'm just wondering, is CD11A, CD11A, and CD6A?
So CD11B plus,
Oh, yeah.
and then CD68. Are they the same markers for microglial cells, or are you asking are they separate?
Like, are they only seeing M1 or is it just the
Yeah, so I'm not going to differentiate those two. So you should know these cell surface markers that we could use for microglia. You should know some of the characteristics and the features of like M1 versus M2 microglia because you have like a nice chart. So whenever you have like a really nice diagram kind of looking at two different things, it's a pretty good idea to make sure you know it. So M1 versus M2 phenotypes, expression of different molecules, like where would you expect to see more arginase, NO production, where would you expect to see like more TNF being produced or interleukins being produced, like the pro-inflammatory ones. Like I'd expect you to know that. So these are markers for microglia. I haven't asked you to identify which one you see more of for M1 versus M2. So if you haven't seen these at some point, make sure that you go back over your notes. I think I can guarantee you that you'll see this in some form or another on your final exam. So microglia is like a good topic to kind of think about. It would be one of those really big topics that I would use for like a short answer question. So like, you've altered like one of these microglial subtypes. What would you expect to see happen? Give me a model system that you might use to show these types of microglial cells. Like you can use an example from class. So, anything else from this lecture? Lecture 7? 7? 7? 7? Lecture 7. Any questions? What is lecture 7? It's a good question. That one I can't tell you the answer to.
Yeah, that is correct.
Lecture 7. Are there any questions from anyone online?
I have a question for lecture 6. When we talked about introducing like in one of the experiments, introducing LPS and then testing for depression.
Yep.
You said it was 2 weeks after surgery. What was the surgery for?
It's so if you remember, most of this was ICV delivery. So you actually have to do the surgery to allow for the cannula to be implanted into the area that you're testing.
So they waited 2 weeks after.
So again, you can use these short forms.
Well, I think there's also in lecture 6. Is N6?
So we're on lecture 7.
Sorry, I just don't know which one's in lecture 6. Is the N6 fed or is it in the actually, never mind. Yeah, I think I remember.
So okay. I don't know if it's lecture 6 or lecture 7. It's okay. We'll figure it out, or you'll figure it out. I don't need to figure it out. You figure it out. So it's probably a good idea to be familiar with like N6, polyunsaturated fatty acids versus like the N3 type. You are going to get the different names of these. You're going to have to know which fall into these different categories. What you're talking about is a little bit different. And what we're talking about is in mammalian systems, we actually do not have this FAT1 transgene. And we use this FAT1 transgene to cause the conversion. So we don't have to worry about giving them or dietarily supplementing these different omega fatty acids. So they convert it.
What does N6, is this naturally in there?
We don't have it. It's a transgene. So it's a transgenic gene.
N6.
No, not N6. So FAT1 is used to convert N6, N3. We don't have that gene. Mammalian systems don't have it. So the FAT1 is something that is normally found in C. elegans. And we take that gene and introduce it into mice, and it allows for the normal conversion to like omega-3 fatty acids that we don't have. So you don't have to worry in that system where you overexpress FAT1 to worry about, like the amount or the type of fatty acid that you're giving is at fault. Sorry, there was a hand up.
To clarify, were you saying that you just do like 576 and fatty acid, we don't produce
neuroscience will still be there.
Correct. Can't tell if you can see on here anymore. Any other questions? So lecture 7. Has anyone finished all the lectures? Like you're done? No. Okay. Okay.
Which one should we focus
Yeah, that's
Last lecture.
the last lecture?
on? Last lecture?
I told you that. I told you that in lecture. Like there's a lot of coverage from the last lecture on the final exam.
Can I ask a question from that lecture, or do you have to go over?
So let's do it really quick. Any questions from lecture 7, 8, or 9? 9, lecture 9. Anything from online, let me know at any point in time.
The opioid pathway.
Yep.
So this confused, where do opioids act on?
All throughout. And any of those synapses that you see there,
Okay,
they're going to be opioidergic.
okay, so this pathway makes you feel pain or not feel pain
So you will be able to block that pain.
using opioids?
Yes.
Okay, so pain signals travel through the zone.
Right, so this is known as the descending pain hypothesis, and we can actually, so these are not people, these just represent like different synapses from your dorsolateral prefrontal cortex, and you have to think a little bit about the other part of it. So we only talk about the descending pathway, so it starts off in the brain and goes down to your spinal cord, right? And so at the level of the spinal cord, we have like the release of opioids from all of these different synapses. And one of the things that we know can potentially happen is that we get pain signals that go up. So you feel pain because it's generated somewhere here first. So something happens in the periphery, it gets put into the central part of the CNS, and then it goes up to your brain. Your brain can then process that, and it can actually decide that I don't want to feel that pain, so it could be through placebo, it could be through like other endogenous release of opioids. So there are, again, potentially different ways to release opioids here, but that's the theory, that this descending pain pathway is a control pathway. So this is the sensory part is the ascending, and then the descending is the control part of it.
So what do you mean when you say that the dorsolateral prefrontal cortex can inhibit pain signals by activating? So isn't the dorsolateral prefrontal cortex already part of the pain pathway? So how is it?
Technically, it's not. So again, if you think back to your neuroanatomy, which I'm very concerned about, every neuroanatomy. So remember how pain goes in? So spinal cord, and then we have like slow pain, fast pain fibers. So we have like the delta pathway or the C fiber pathways. These all go up into the prefrontal cortex, right? So S1, that's your cortex. This also sends projections to the dorsolateral prefrontal cortex. This is anterior or in front of S1. This is where you integrate pain signals. And that's where the CNS, your brain starts to integrate it. And then it sends fibers to things like the dorsolateral, it also sends descending pathways. So in theory, you could also have something going from here to here. But again, don't get too confused on this. Like this is where you get the ascending part, stops here, sends projections here, also sends projections back down to the spinal cord. But this is where you can inhibit the pain. You can block pain sensation. But they're not the same. They're not the same. I may have to go back and revisit at some point like what I assume everyone's background. It may have been a while since you've had that in physiology, right?
Yeah, some of us don't have it till later, so we'll have it after.
Yep, okay, perfectly fine. So thanks for the clarification. I'll make sure we do that in the future.
I think it might be in lecture 8, but
Okay, lecture 8.
it
So
might be 9. I'm not sure.
okay, one of those. Okay.
The experiment, speed-limit experiment with PGC1 alpha.
Yep.
How did I just was a little unclear as to like how you found that or how you hypothesized that PGC1 alpha is a co-activator of FMDC5? Because wasn't he looking at FMDC5 in muscles and then he said like PGC1 alpha is in the brain?
No. So some of that it's a little bit complicated, but the way that I approached kind of his background to it, his original rationale for looking at this is again, I think I talked about it, he had like a PGC1 alpha knockout animal. And then when he had like this knockout animal, he would actually see that there were some malformations in the brain. So without this, the brain actually didn't look like it normally should, and like the ventricles were bigger, there were some areas that had deteriorated. And so he made this link because PGC1 alpha is not only expressed in the brain, it's expressed elsewhere, that this factor is important, but maybe like this links with like exercise. So his idea was to use this observation that he had first with the PGC1 alpha knockout animals that impacted the brain. And because his interest was on the effect of exercise on the brain, that maybe this is that missing factor. And then when he looks into muscle, we know that FNDC5 PGC1 alpha is part of that process. So his knockout was specifically to the brain, and then he started to reverse engineer like what might activate PGC1 alpha in the brain. Okay. Is there a question online?
Interwebs. Yeah.
The interwebs.
A question about hawk speed. So someone just wants to know that if the only thing we need to know about hawk speed is that its expression in NG cells is important to prevent anxiety, OCD anxiety.
Which lecture is that from?
I think that might be 7.
We're going in order.
No, we're not. We're jumping around, but that's okay. So you're asking about hawk speed.
Yeah, hawk speed and the microglial precursor. Or I guess, and actually mature microglial cells, it should be, right, like 30% of them.
Yes, in small proportion of these cells, we will have like hawk speed. I would say that's probably enough information. So whatever I covered in lecture, I can also just indicate, since you've asked that question, that this molecule is probably one that you want to go back and revisit now that you've reminded me. So who was that person online?
It was one Andrea Mackenzie.
Good job, Andrea. Good job. Good question. Good question on hawk speed. If I didn't clear that up, let me know. So yes, good molecule to go back and review. So I know we didn't talk about it very long, but I did talk about this in the new context of the new theory.
Actually, someone else just mentioned after that question that you also, the one other thing you mentioned with the NG cells was the CSF1 receptors that went along with the hawk speed.
Right. Okay, yes.
I'm not sure if you mentioned this one, the NEOAs.
Yep.
I think it's a placebo. You said higher NEOAs lead to lower placebo responses. So that means like they don't have a placebo, right?
That's correct.
Okay. So how does that work? Having like more breakdown of neurotransmitters into
Right, so the short answer is we're not 100% sure how that works. We do know that some neurotransmitters have more of an effect on the placebo response than others, but we're not 100% sure. We do know that they are better placebo responders though.
Okay, so since we said that higher NEOAs lead to lower placebo responses and it's correlated with the polymorphism, can we say the other way around? So like lower NEOAs lead to higher placebo responses with TT polymorphism?
I would say higher being like you would expect to see placebo responses in individuals, yes. Yes.
So it works the other way.
Yes.
Yeah, just adding on to that, it says that they're correlated with the GG polymorphism. So the GG polymorphism giving you, making you more likely to fall for placebo is correlated with the MAO, making you less likely to fall for placebo.
Can you read me the whole sentence?
Individuals with higher MAO activity have lower placebo responses that were correlated with the GG polymorphism.
That's correct. So GG leads to higher activity, which leads to changes in the placebo response.
Okay, and then besides that MAO activity with EP polymorphism, does that mean that everyone with higher MAO activity has an EP polymorphism?
Not necessarily, no. There are other polymorphisms that are also associated with it, but they don't all lead to improvements in placebo responses. Nor does having like MAO inhibitors. So if you can have like MAO inhibitors to treat depression, for example, doesn't mean that you're going to alter like the placebo responses.
For this purpose of EP polymorphism, so heterozygous, can be treated the same as increased activity in MAO.
I think the better way to approach it is always stick with the homozygous because that's an easier interpretation. So like GG versus like TT. Because GT is like, it means they're heterozygous. They have like one copy of one copy of the other. It becomes really confusing. And it's similar to like having one copy of the long versus the short form of the 5-HT-LLPR.
Okay, I guess I'm just trying to understand why it's written after MAO activity, but I was trying to figure out.
How about we try to figure that out offline? So let me just, because it's like 2:15, let me get through like the rest of the questions and then I'll have a sit down with you and answer them. Yeah. Other questions? So 8, 9, 9, 10, I forget what was 10. What was 10?
The most important one.
Most important.
Is that one is at the last lecture.
Yeah.
Okay. So what did I cover in the last lecture?
Good question.
Oh yeah, meditation.
No, there's a lot in that last lecture. It may not seem like there's a lot because we finished in an hour and a half, but there's a lot in that lecture.
That's not scary at all. It's cool.
No, there's a lot in that lecture. So there's a lot in that lecture about inhibitory neurons. There's a lot in that lecture about so do you remember these? How much do you remember without looking at your notes?
They're only found in grade 8s and humans.
Humans, something else?
Yeah, it's not
No.
in mice, so we can't use them.
Nope, something more. Spindle, right. So these are like spindle shaped, important, right? They are thought to be like unique to us, maybe higher apes, maybe whales, but these are also spindle shaped. So you should know that. These inhibitory neurons, like they come in different flavors, different layers, so you know, okay, I'm assuming you know that we have like 6 different cortical layers, and you should know that, I'm so sorry. You know that there are different output zones in these different layers. And you should probably go back and review, as I told you during lecture, what these different types of inhibitory neurons are. So our chandelier neurons, inhibitory neurons,
Yes.
are von Economo. They're not. They're excitatory, but we didn't actually cover that, but they're not classified as inhibitory. So chandeliers are other examples. Basket cells.
Double bouquet.
Yep, so the double bouquet. Do you know what layers in the cortex they are? Because that's
That's my question. It's like a little bit difficult to see because sometimes they go like halfway up between 4 and 5. I'm wondering, how's this like
But can you tell the difference between layer 3 and layer 4, 5? Yes, you can.
3 and 4, 5?
Yep.
Yeah.
Yep.
So it's not going to be like, is it found in 3 or 4?
If you were asked, like, is like a chandelier neuron found in like layer 4, you could probably make the argument one way or the other, but you wouldn't be able to do that with like, is it found in layer 3?
Okay.
Right.
So it's like, it's gotta be obvious.
So if you're not 100% sure, you should always like the layer that you remember, go for it.
Can we mention like
No, because it may just be a multiple choice. You won't be able to mention. If it's a short answer, you can. Sure. If it's a short answer, 1-line answer, yes, you can go from either one. It's either layer 4 or layer 5. I'd be okay with that. But if it's multiple choice, like you go with the answer that you remember the best.
can we do 4/5?
Yes. You can do 4-5. You You can do 4 and 5. You can do 4 and or 5. You can do 4 or 5. Any of those are acceptable, yes.
Really quick, like tau-retin and
Bindin.
tau-bindin and all of those, are those proteins associated with it? I don't really know any of that.
Yes.
So I think they are proteins associated with the nerve, but
Yes, but okay. Okay.
I thought these neurons express these proteins. Isn't that the
So okay. All I can say is okay. If some of you go on to take like CJH, where we use like different driver lines, which is a molecular biological technique. So let me make this really clear, so I'm sure that we can see this. So we talk about different types of inhibitory neurons, right? So when we talk about inhibitory neurons, we can identify them by their shape. So one way we can do it is to classify them based on morphology, which is what we just did over the last 2 minutes. We looked at basket cells, we looked at double bouquet cells, we looked at chandelier cells. And if you look at their staining patterns, they are unique. So if we see those, especially at those different layers, we have a pretty good guess that they're inhibitory neurons, right? Another way we can classify them is through like a molecular technique. If they're inhibitory, then in theory, they should also express specific proteins that are found in inhibitory neurons. And some of those different molecules are things like tau-retin, and in this case, things like calbindin. Like those are different ways to identify inhibitory neurons in that slide. There are other ways too, like GAD or glutamic acid decarboxylase. But these are markers for inhibitory neurons. I don't know if that was clear in your lecture, but it's something that you should make sure that you are aware of. Yes.
Just a quick question from the group. How they're wondering
Which
how
group? Different group than them?
Google Doc.
No,
Never mind.
Google Doc.
Um,
I'm on YouTube.
They were wondering how much of the pancreatic cancer do you need to
Sorry, how much of a what?
Pancreatic cancer,
Steve Jobs.
yeah.
You get, I would say you don't have to know about the ways in which the surgical removal, but you will get like a multiple choice test question for sure on like the pancreatic cancer lecture. I think also like a short answer question. So it's probably worthwhile going over unless you don't care about marks.
Thank you.
That's the last.
Yep.
So carvalhoin is a marker for also?
Yep, it's another marker.
So 3 markers.
Yep.
Yeah.
So again, like for me, I would kind of assume that you guys have all encountered them before. Again, an assumption and something that I need to go back and revisit. Like you, CAD 68. You guys have heard of it, but you may not all know of it. So I kind of make the assumption that things like parvalbumin, because if you've taken like a molecular course, we often use like parvalbumin Cree driver lines. These are inhibitory neurons. Inhibitory neurons, inhibitory neurons. Any other questions?
I'm not sure where the bomb neuron shows up.
Sorry?
Right 8% humans.
No, like, I mean,
Cortex.
like, how does it apply to the rest of it? I don't really.
So that whole lecture was really trying to get you to think. So we use mouse models, and what that part of the lecture got you to think about is our brains, the circuits look very similar, right? So the circuit that we have between the hippocampus and the cortex, or the amygdala and the cortex, the circuit that we have between the VTA nucleus accumbens, that circuit is pretty similar in mice versus humans. But is a mouse brain the same as a human brain? Because that's important in our discussion about whether or not we can study like higher level behaviors. So placebo effect is like a higher level behavior because it's really hard to kind of come up with a model system where you can test for a belief system like placebo. They have to believe that the injection they're getting is going to make it less painful. But we went through a model system where we could do that. We also identified the circuit that allows us to go back and use things like naloxone to block it. We know that if parts of the prefrontal cortex deteriorate in Alzheimer's disease, that we lose that placebo effect. So we know the circuit, and we know that it works in mice and it works in humans. But then when we start going even higher, so if we are thinking about meditation, right? So meditation, we can't really test meditation outside of humans easily. So that got into, are human brains the same as mouse brains? Von Economo neurons are unique to human brains. We don't see them in mice. So that got us to think a little bit about it. But the study was around meditation. And in meditation, can we use like different rhythmic activity in different areas of the brain to make it look like they've meditated? And is that enough for them to have a reduction in stress and anxiety? That was the whole point of the Von Economo. Our brains are different, but the circuits are enough to allow us to replicate higher order behaviors like meditation. Any other questions? Any other questions online? Not from YouTube? Google Doc people? No? Any other questions in general? Lecture 10, 9, 8, open? Just open, random? Yes?
Just back to lecture 5. So you know,
You're killing me. Yes, okay.
So you know we said for the shorter alleles, there's less of the study transporter. So how does that relate? Like that means that there's less serotonin. I mean, there's gonna be more serotonin, right? In the synapses. I'm kind of confused.
Yes, well, it's a confusing field, and in fact, this year is the first year, I think, that I've actually shown you this on one of the other slides. So, lecture 5, and I'll just do lecture 5/6. So, your theory, if I can kind of wrap it in that, is that if you're depressed, you're given like a selected serotonin reuptake inhibitor that will increase the levels of serotonin, hopefully reversing depression. And so why is the opposite true, right? So how is it possible that if you have like a reduction in transporters, you would expect more serotonin around? That's your line of reasoning, right? Okay. So there's a couple of things. So one, before we get to that, if you look at the next slide, which I think was the one that you were asking about for the trans fatty acids, we talk about like the effect of diet and other things. Buried in that one slide, and I mentioned it in class. Do you remember what this is?
Selective serotonin reuptake inhibitor.
Do you remember that one? That also causes depression, or this can also like be affected in depression and can also reverse depression. So we don't really know. So that's the first answer. So SSRIs might help with depression, but so can SSREs. So that's the one thing, and we don't understand it.
So
But
it's not just that like more serotonin means less.
Not always. That's what we've learned over the last 20 years. So this can also affect depression, so can SSRIs. So we don't really understand it. So going back though, what the way that these authors are kind of explaining that pathway, and you don't need to explain it in the same way, is that when we look at these different groups of neurons, and we're looking at the levels of serotonin here, do you remember that this was a prospective study? So they followed people over time. So we start our study today, then I contact you like 10 years from now, hey, how's life been? So it's a prospective study. They start off when they're really young and they want to know, like 15 years from now, everyone in this class, how many of you have had like an episode of depression? 0. Okay, great. That's awesome. That's what we would hope. But the reality is when you go back and you take a look, if you look at these different individuals, they also had to have one other thing. So it's not just the gene. What else did they have to have? They had to have a stressful event. So most of the time you're kind of like, okay, no problem. Things are okay. And you don't really have to worry about the fact that you have like low transport levels of this reuptake. So you can get by on just the basal level of serotonin under most conditions. And then something stressful hits you. And then things kind of fall off the rails. And one of the reasons why they think that this happens, and again, it's just a hypothesis that no one's been able to address one way or the other. So under normal conditions, like you release serotonin, some of it gets degraded, some of it gets taken back up by serotonin reuptake transporters, right? And we know that the only thing that's different is they have less of these transporters. But under normal conditions, and again, it's a terrible analogy, but I hope that it makes sense to you. Can you live with like one kidney? You could live with like one kidney, and in fact, you could be sitting beside someone that has like one kidney, no problem. But when they're physiologically stressed by like drinking something too much of something, or they have other issues, that's when we know that there's something wrong. And so for these individuals, they get by with like the basal level of serotonin all the time. But then there's a stressful situation, they've like depleted they've released all of their serotonin. But then they have nothing left because they haven't been recycling it as efficiently and as effectively. And so their levels of serotonin now, after the stressful event, are depleted, gone, and they have no way to replenish it. So their levels of neurotransmitter are indeed like lowered. That's their hypothesis. So they've just had less, but they've been able to get by on that until like a stressful event. All of their systems, including like their norepinephrine, serotonergic, all those systems get hyperactivated during stress. They release all their neurotransmitters. They don't have an effective way to reuptake those neurotransmitters. These neurons are now facing like a crisis and not having enough. Again, a hypothesis. Hasn't been proven one way or the other. Okay, any other questions? Do I really look like that? Wow. Okay. So, shall we try this? Do you have any other content-related questions? By the way, will I ask you a question related to any of the meditation slides?
Yes. Yeah,
Um,
you said it was super important.
Well, I think you should probably go through like the bullet points on the slides related to meditation, how we look at the effects of meditation, what areas of the brain, the different techniques that we use to measure like stress, etc. So those would be good things to go over. Okay, so going back, so for those of you who came a little bit later, and you guys were able to type this question out. So this is an example of like a sample-ish question, but I'm using this to kind of illustrate a couple of points. So you're going to have something similar. It could be like you're knocking something down, you're affecting a circuit, you're overexpressing something. So this is like one example of one type of question that I very often ask. So here you're overexpressing AKT kinase. So right away that becomes one of the things that you should, like what do I remember about AKT kinase in neurons? So again, this kind of takes away from like, do I have to worry about M1 and M2? You don't, so it's neurons. And hypothesizes that overexpressing AKT kinase will impact anxiety. So again, you're thinking, okay, what do I remember about anxiety? So in your answer, you should be discussing like which neurons they would target for overexpression. the different behavioral tests and then their predicted outcomes for 10 marks. So does anyone want to get us started? So I'll take like whatever answers you have, then once you're happy with the different parts of the answer, then I'll go back and start marking it. So someone want to get us started? Nobody wants to. Yes, Sam.
So overexpression of AKT kinase will block apoptotic pathways. And then that impact on anxiety would enable neurogenesis.
Okay, so again, one of the things that I mentioned is like before you actually get started on the question, like just write stuff up at the top where you don't have to worry about things in the margins. So your key thoughts are is that maybe we can look at neurogenesis pathways, possible,
Survival.
and AKT will increase survival. This might somehow impact things like or decrease like anxiety. Is that your hypothesis?
Yeah.
Okay, so that's okay. Anyone want to expand on that, or anyone want different tacts, different pathway, different ideas? There are like always multiple answers that I would take for each of these short answer questions. Generally, there's one that I'm looking for, but I will also like adapt. Anyone? Sam, they're leaving me like hanging me up there.
Do you want to expand on like what that does or
You can-
another pathway?
So this won't be your final answer.
No, there's the parts.
Okay. So what else were you thinking about for some of the other parts?
So for neurons, we just target the ones that like we see adult neurogenesis in. So like maybe like dentate gyrus, subgranular zone, or campus. And for the different tests, we'll talk about like for swim tests. For anxiety, wasn't it the more isolation ones, like the bigger rat, just a smaller rat, like the mental helplessness one,
Mazes.
the mazes.
Was that the whole thing?
Yeah,
Zero
yeah.
maze.
What's the
Like
other one? The bigger ones, the smaller ones. That's pretty.
That's social defeat.
Is that anxiety or depression?
I think it was.
I thought it was anxiety.
That one was the one where it could be both,
Maybe
there was like a
both, yeah.
controversy.
Yeah. But one of like some of those when you get tests. No, you don't have to do
Which three are you gonna pick?
the maze test, the
Zero maze? Yeah.
The open
So-
field
so-
test.
So Sam, like if you put like maze test, you would have got zero. So sorry to say, you might have that in your head like what that means, but there's like a Morris water maze. That's a totally different test. You might have it sort of as an elevated plus maze in your head. Fine, I'd take that, but I can't tell like what you're thinking. So like a maze test, zero. Like I know you think, oh, half a mark, but honestly I can't tell like what if you know it or don't know. So pick one of those.
the zero maze.
Okay, so zero maze. So and so let's pick three different ones.
Like
Like
a
dark
open field test.
Zero.
They love Texas.
Yeah. That, okay.
You could also use like social interaction, social approach. I don't care. You can use either term. Social approach, interaction. So okay. Boy, just realized if I wrote like that and I couldn't read it, you wouldn't get words. But I can read my own writing. So if you can't, then you can go ahead and deduct points. So okay. And what's the predicted outcome if you overexpress AKT?
Less anxiety,
less
so.
Okay, so here is where you need to interpret what I mean by this, like, as close as possible. So 3 different behavioral tests and their predicted outcomes. So what you should be describing, like, what I would expect to see.
More in the open regions for the zero maze. More in the
So-
center for open field. More in the light for light and dark.
And more social.
More social.
More social, yeah.
If they have social anxiety.
So again, just making sure that with overexpression of AKT kinase, that you would see this. Okay. So I'll just make sure that each of these is correct, so more time on one. Sorry, do you know how this one works?
Yeah, more with the,
Interact more.
with the.
Interacts more with,
With another mouse.
With the, instead of the inanimate objects, with the.
right, so you actually see like the amount of time it's spending with one versus the other. So
Almost as difficult because like it's skeleton has to measure.
it's all automated.
Okay.
Literally, you put them in a box. There's a little computer. There's a little camera that tracks the movement and actually sees like how much time it's spending. So it's actually not very complicated. So this one here, like the way that you had assigned that one, like you would have only gotten like half the marks because I need to know that you understand not only the tests, so you would have gotten like marks for the tests. So generally, like how I mark them is I take a look at about, I just randomly do them. So just again to let you know, I try to eliminate as much bias as I can. So the first round of marking, I go from A through Z, and then I go from Z back through A, and then I go A through Z, so that I take a look. I also take a look to see if like the marks are getting like higher, lower as I start marking. So if I'm finding that, oh, by the time I get to Z, it turns out that I'm marking harder and harder, then I'll go back and see like where that process might have occurred. I try to mark like all the questions like in a single sitting wherever I possibly can. So just like, okay, 26, like that's all going to be done over the next day, and then 27 will be the other day. But generally speaking, I take a look at about 10 exam papers that I pull randomly, and I'll take a look like, oh, how did people try to answer this question? Then I'll sort of sit down and come up with like an appropriate answer key. So I have one in my head, but based on like how the class answers it, like it might have to change. Like, oh wow, for some reason like no one is talking about the dentate gyres. Like everyone's picking the amygdala, and should I look at what that means? Or they're all picking like these monoconomal neurons for some reason. So like, is there a reason for that? So generally, like I'll try to map out like an answer key based on like the 10 random exams that I pull, and then I kind of sit down and go through the rest of them. So you would have gotten part mark. So these are all good. So I like those. And you might be thinking, oh, so 3 different tests going to have 3 different marks, and then each of these are going to have like 1 mark each. Don't make that assumption because it may not be true. It may turn out that way because like that's the way everyone's been doing it, and it looks like everyone's gotten really good responses. Then I'll take that. But it may turn out that more people spent time like on describing the neural circuit. They spent less time on this, and so I'm not going to put as heavy an emphasis. And it may be 3 and 3, or it may be like, oh, well, I'll take like 2 marks here, and then most people spend so much time up here that I'm going to weight this more heavily. So like, don't make any assumptions because it may be wrong. So for this one though, just to go back, so the social approach, so all of these would be correct. So let's assume for the time being that each of these is worth 1 mark. Just again as an example. So the way that you described it verbally to me, so more time in the open part of the maze, perfect. I would say that. Overexpression of AKT kinase, so you would have gotten a mark there. So more time in the light compartment versus the dark compartment. So again, I would say you get a mark there. And let's assume for the time being that you didn't do this and you just wrote 3. So for like the social approach, if you had spent more time with the animal, that to me wouldn't be a complete answer. So more time with like a different animal or mouse, rat, like I'll take all of those, versus like an inanimate object. Like you need to finish that. So that would be like a half mark. So I am very particular about like you just have to be really clear, and that's why I suggest like mapping this all out. So going back, like would you necessarily want to have like an increase in the dentate gyrus and the sorry, not the substantiator, the subgranular neurons? Would that be useful? Why?
Because that's the hotspot for neurogenesis.
Okay, but how does neurogenesis tie into anxiety? Like that one key link. Sorry? It would increase BDNF. Okay, not sure what you mean by that. Yes, Sam?
So there's no like LTP and like plastics, like plasticity and stuff. Like when you have more neurogenesis in those areas, like you increase plasticity and like it's better for learning and memory, which anxiety kind of targets sometimes.
This one is an example of a fairly challenging one. So again, let me just go back. So if you're gonna start off this way, so like this might affect like neurogenesis and survival of like newly born neurons. It might affect newly born neurons specifically in this. So I love that answer. So very specific, very logical. You're looking at AKT, you know what pathway it might be affecting, and just mentioning how this might tie into anxiety. So you might just want to work in that they might be connecting to like the amygdala, which is normally where you see fear responses, or you might even go again. You need to kind of expand out. Do you remember like the area that is involved in like self-control? So you could make like a hypothesis that maybe through increased neurogenesis you get better connectivity to the ACC to reduce like anxiety. But you need to kind of tie all of that up. So again, it was a great, brilliant answer, and we only have like part of the question out here, so you can get the more fully developed one. So on that, sort of based on like what we just discussed, you would have got about a 7.5-ish. You need some more details to kind of link these different things up to anxiety, to be a bit more specific on the different tests that you're implicating. And again, somewhere between a 7.5 to 8.
Do you want us to make hypotheses like that?
You'd be asked.
Okay.
So, or if you're asked about like anxiety and these different structures and how they relate to anxiety, you'd have to kind of hypothesize. And by the way, you're free to use any experiments that were talked about in class. You don't have to make up a brand new one. Students often ask, like, do you want me to talk about something brand new or something that we talked about in class? Class is fine. And this gets back into like specific neurons that we looked at related to anxiety. So anterior cingulate cortex, making connections to the anterior cingulate cortex. You could talk about all sorts of things, but here you're locked in. We're not looking at microglial cells. We're not looking at different factors. We're looking at neurons. So relate them all back to neurons. Anything else? Any other questions? Nothing? Any other random questions? Random questions from people online?
If we have other questions, do you have any recommendations for asking? Or should we just ask for other classmates?
No, don't ask your other classmates. I'm only saying that because I know students get very anxious, and so like anxiety is contagious, since we're talking about anxiety. So I think what I might try to do, what time is your exam on Thursday?
2 to 4.
Why did I hear different times?
Two. It starts at 2 to 4.
2 to 4, okay. So, I think, Thursday. So, I think this should still be okay. So, if you have any questions between 10 and 1, like I'll be in my office, you can come and ask. That means also like you can email it during this time and I'll also answer. I prefer it on discussion because I don't want to have to repeat the same answer over and over again, but you can drop by Thursday 10 to 1 to ask in person or put it on discussions and I'll answer that. So I don't know if that makes it worse or better. But Kiza, you have a different suggestion?
No, I have a question. It may
What
be
does
a
that,
little this
what does that
question.
mean?
I'm hesitant to
Not
ask it, but I
sure.
want to ask it.
Yeah,
You see, for the first part of the question, how you're able to connect all these different components of lectures together, I find that's the hardest part. Like, do you have any advice with how to make sure we have a big picture?
To be honest, we talked about that a little earlier on. So I mentioned to your classmates like how you should be looking at keywords. Many of you do this already. So you look at AKT kinase. Where have I seen that before? And you're looking at like neurons. And so I think this is how, for example, Sam got to this neurogenesis, effect on neurogenesis. We've seen AKT, we've seen this pathway. And then you look at other words like anxiety. So like these are linked. So like anxiety tests, groups of neurons. Neurons are always the ones that, the circuits that the neurons form are always related to behavior. So that's hopefully, yeah. I'd say again, for most of you, and I don't think it's any surprise, you should probably look at the microglial cells like the M1 versus M2 pathways or cell identifiers. I think, again, it's a pretty good idea for detail if you haven't done it in a while. So like N3, N6, and we're talking about polyunsaturated fatty acids. Make sure that you're good with that. If there are any experimental details that you're not clear on, then you should probably use that approach as well, Vikiza. Like just go through like the big experiments. Any other questions?
Thank you so much, Professor.
Sure. Right. Well, just manage the stress. Take the stress down. It's really hard to kind of manage it, but it's really important because I'm looking for detail, but I'm hoping you have a better idea of what I mean by detail and what I mean by like integrating different topics or different things from different lectures. And to be honest, I didn't make it as interconnected as I could have. Like as I was talking to you today, like there are a couple of questions that came up. Like, hey, that would have been an awesomely hard question. But like there are enough things kind of linking some lectures, and not all the questions are like that, so you don't have to worry too much. So just try your best. It'll be okay. I'll be over at 4 o'clock.
And then freedom.
And then freedom for many of you. And no online questions? No online questions? Do you guys want to try to book a space again like last time, or you guys are just good at just dropping in? Should we book a space?
Maybe book a space.
Alright.
If possible. You can remember like this. Yeah. Maybe the same room.
We'll see if it's available, if they have a key. Alright. Okay, so alright, thanks everyone.
Thank you, Professor Hughes.
Yeah, and I know you're I know some of your marks are starting to come up, so participation marks will be up sometime on I think sometime on Thursday evening after your two tests. So like the participation, as long as you were registered, you attended at least 2 classes and there was some kind of interaction, like the class average is like 4.75, lots of 5s. Only 273 students out of 303 actually registered though for autonomy. And about 5 of them, I know because they don't carry a laptop, but everyone else, like, where did they go? Why did they not even register for autonomy?
But it's like a free.
It's like a free program. I don't know.
There's issues like the site, like they want, like you register on your phone, there's issues with the first.
So as long as you're registered, as long as I could see that you were interacting with at least 2 classes, like they got 5 out of 5. But again, there are some students where no registration, no interaction. And this does not include students that indicated like I write my notes out by hand, which I already accommodated like in other ways. So as long as you did that, it's like 5 out of 5. But the average is not 5 out of 5 because if you didn't register, like I can't do this.
Thank you.
Alright.
I just wanted to say that I noticed this. Did you realize the AKT gene discovery was a U of T discovery?